This is Holy Cross Mark for component 2, and it's Mark 4. Read the following passage, blah blah blah, all about shark fin soup and DNA, but it's certain things you should recognise in here as you go along. You should know about DNA, base sequences. It's throwing genetic fingerprinting in there, and then it talks about primers, which you might not know a lot about, but it tells you what they are. It says they are single-stranded, so like half a DNA, complementary to other bits of DNA that presumably the shark has. And all the things they're throwing in there: polymerase chain reaction, and then they're just explaining what they do with the DNA. So you're kind of ready in this question for stuff about genetic fingerprinting, complementary kind of binding of the DNA, and PCR. That's the sort of thing you're expecting just from reading that. And then it's a nice little warm-up, is this. Explain why the DNA for each species of shark shows differences in the base sequence. So why the different species have different DNA? Well, they're different, aren't they? So different species have got different genes. For example, like you've got different genes to a dog. It's got a much better sense of smell than you, and presumably genes have something to do with that. So different genes in different species, and of course differences in the base sequence. Well, it's the base sequence that determines the protein that and then the different sort of characteristics. So different genes in different species that produce different characteristics. And these are from different proteins. So different proteins produced, or different enzymes perhaps, give the different sharks different characteristics, and those are from different genes. And of course, it's the DNA sequence in genes. How did all of these come about? So why are they actually different in the first place? Well, mutations cause the changes from a common shark ancestor that had a certain DNA sequence. So you just need kind of two of those things in there. Each primer, remember primer, so that's all. And here's a double-stranded DNA. Okay, a primer is just half of that. So it's got exposed bases instead of bases that are hydrogen bonded onto the other strand. So exposed bases, that could match up with another strand. Will it match up to that strand, or will it match up to that strand? Well, it depends what basis we've got. And so why a particular primer will only bind to the DNA of one species? Well, if we've got ATGC there, and this one has got AACA, and this one has got TACG, you can imagine if this one bashes on to this, it's not going to stick together. It's not going to form hydrogen bonds. There's only that A there that could possibly make a hydrogen bond with a T, and so it's not going to attach. Whereas this strand here, you can imagine it carrying on. All of those bases are complementary, so that strand there would like nothing better than to attach on there. Okay, So that's why a particular primer will only bind to the DNA of one species. So that's species 1, this is species 2, and so species 2 is the one it'll bind to in this little example. So our primer has a DNA sequence, so a different DNA sequence, and that's specific, and complementary to, another strand. So the DNA is specific, complementary base pairing to another strand. That sort of idea there. The length of the replicated DNA fragment is different for each species. Okay. Explain why this is important. So, remember, it's telling us about genetic fingerprinting. What do they do? DNA is run in an electrophoresis gel, and that produces a band. So, if you remember a gel with a kind of a solution inside, a buffer solution, and if we put DNA in little wells, you've probably done this or had a look at this in class, and the DNA will move when the current is switched on, and it'll move towards the positive because DNA is slightly negative. And it tells us that we get one band, because we get one primer attached to a specific DNA sequence. We're going to get one band, and it's telling us here that the length of this fragment is different for each species. So species A with a little fragment might go up there, species B with a medium fragment might be there, and species C with a large fragment might not go as far, because smaller fragments move further up the gel, and that's going to allow us to identify these species. So let's say electrophoresis separates the DNA, and they can be identified by their position on the gel, and then we can just say, well, why are they different? So smaller fragments move further, or larger fragments don't move as far. So that's how we can identify the species. When we say genetic fingerprinting, so the next question, normally get a series of bands, so you're probably used to these, these sorts of bands. And the criminal, who's the criminal, and that sort of thing. But actually, in this situation, there's only one band. And explain the reasons for the difference in the number of rungs produced. So why is this only single as opposed to normally it's got lots of bands? Well, when we do normal genetic fingerprint, we take our length of DNA, and this gets cut up in all sorts of different places, and it depends on the short tandem repeats and in the introns and so on. So you get lots of fragments, whereas in this technique they're looking for one gene, and the primer will attach to one section of DNA, on one of these fragments. So we only get one band. So we just need to put that into we make sure we know what we're talking about. So the difference, we can talk about either. So let's talk about right in red, conventional DNA fingerprinting has many lengths of DNA to separate. So there's lots of fragments of different lengths, whereas the new type, I'll just put this in brackets, new, only one length. And each rung of this is corresponds to a specific length. So each rung corresponds to a specific length. So, and as with our sharks, we're only using one fragment. Well, that's only one length, so it'll only go to one position. And then part C, 6 marks, so that's 2/3 of an essay. 2/3 of an essay just to talk about polymerase chain reaction. This is a gift. Okay, so all it is a polymerase chain reaction. So we start off with one bit of DNA and we're going to separate it using heat into 2, and then we're going to add primers and then add nucleotides, cool it down, and then we get 2 bits of DNA. And then we're going to do the same thing with those 2. So we're going to separate that one, we're going to separate that one, we're going to add new blah blah blah, and then before you know it you've got that much DNA, and so on and so on. We need enzymes and we need temperatures and that sort of thing. So that's the description that we're going to give. So what goes on in what order? So we are going to heat the DNA. That will break the hydrogen bonds to separate the strands. This is the bit you're probably a little bit more ropey about, is adding the primers. We need to add primers. We're not as clever as cells that are doing DNA replication with a whole bunch of enzymes. We need to sort of cheat a bit, so we're going to add primers, small lengths of DNA that are complementary to part of this DNA, and then that's going to provide an attachment point for DNA polymerase. So we add the primers. We also need the bits of DNA to build the strand of DNA. So what are the bits of DNA called? Those are nucleotides. So add nucleotides. And then when we cool this down, hydrogen bonds will form again. So cool down. You probably know the temperatures. I can't remember the temperatures. Cool down, and that will this allows binding of primers or nucleotides, and they'll form hydrogen bonds. And what's the enzyme that zips it all up, forms those covalent bonds, phosphodiester bonds? So that's a DNA polymerase, and that forms bonds between nucleotides. That is possibly not clear enough. We could say bonds making the sugar phosphate backbone. That's clearer. That's saying it's not the hydrogen bonds, it's the kind of covalent bonds, the phosphodiester bonds they're called. And of course, I did this, once, twice. But if you did this many times, you'd end up, not enough ink to write all the DNA strands. So repeat the cycle many times to get lots of DNA. So even if some of the other questions you had a little bit of difficulty with, your 6 marks there, dead straightforward.